All right, time for our last topic in Chapter 5. This is 5.4 on cladistics. Okay, so we previously talked about those taxa, kingdom, family, class, order, family, genus, species. Okay, so how are organisms placed into those groups? Well, we can use multiple characteristics. We can use morphology, which is the shape of something's body or a body part, like the shape of a bird beak. We can use anatomical structures, like monocots have flower petals in groups of 3, and dicots have flower petals in groups of 4 and 5. So anatomical things, or cytology, cyto meaning cell, like what the structures and functions of their cells look like. If we dive a little deeper, we can take a closer look at some of the internal, like intercellular things that make organisms unique, like phytochemistry. So phyto, we've talked about that before, like photosynthesis in plants. And this is an analysis of special chemicals that plants make to defend themselves from insects. And those are going to be pretty unique to each group of plants. We also know that chromosome number is a defining characteristic. So normal humans have 46 chromosomes in each of their somatic cells. So anytime we see species with the same or similar chromosome numbers, that might tell us that they belong into a similar group or that they're closely related. And the last one, which we've talked about quite a bit, are molecular comparisons. So this is either when we can compare the amino acid sequence of a protein or the DNA sequence of nucleotides and see how closely related two things are. So probably by the time that some of you are biology teachers, you're not going to be teaching your students about kingdom, phylum, class, order, family, genus, species. Okay, that is going by the wayside for a new way to classify things called cladistics. And this is a system of classification that groups organisms based on their evolutionary history. Okay, so as organisms evolve and then diverge from common ancestors, we can literally trace the development of different features and put things into groups called clades. Okay, so again, this relies on knowing common ancestry. And so to figure out the common ancestry, of course, we can use DNA sequences and other things, but we can also look at how primitive and derived traits that different organisms possess. Okay, so primitive traits are exactly what they sound like. Those are characteristics that are old, okay, and that have evolved very early on in the history of that group of organisms being studied. So for example, in our groups of plants, vascular tissue evolved very early on, okay, and so we consider that to be a primitive trait. So every organism that branches off in this cladogram after that appearance of vascular tissue, again, is vascular. So all these guys have vascular tissue. That is a primitive trait. Only the mosses, the bryophytes, don't. Derived traits are things that have the same structure and function, but they evolved a lot more recently. So for example, okay, flowers only evolved very recently in the evolutionary history of plants. Okay, and so those are going to affect a much smaller group of organisms. Okay, so I keep mentioning this word clade. What is a clade? So we're going to talk a lot about phylogeny, and phylogeny is kind of a tracing of an evolutionary history of a group of organisms, and a clade is a monophyletic group, okay, mono meaning one. So it's a singular group composed of the most recent common ancestor, so only the most recent one, and all of the species that have descended from that common ancestor. So organisms are placed in clades because they share characteristics. And the smaller your clade, okay, the less organisms you're going to include, but the more characteristics that they'll have in common. Okay, so for example, if I wanted to make a clade that included vascular plants, okay, I would only be including these individuals, okay, conifers, angiosperms, and their common ancestor. However, using the same diagram, I could make a different clade. Okay, I could make a clade of all land plants. And so if I wanted to make a land plant clade, again, that's going to include more organisms and a more ancient recent common ancestor because that trait applies to more of these branches. So one of the things that we keep seeming to come back to in IB Bio is this universality of the genetic code. Okay, so we often say that the genetic code is universal. And so what does that mean? Well, that means that pretty much every organism on Earth has DNA that functions in the same way. It's some arrangement of A's, T's, G's, and C's. Okay, and when we transcribe and translate that DNA, okay, and group it into groups of 3 codons at a time, the same codons, like AUG, is going to make methionine. It doesn't matter whether you are a bacteria or some kind of thermophile or a fish or a human. It doesn't matter. Now, the only way to explain how all organisms on Earth got the same type of DNA and makes the same amino acids is if they all originate from one single common ancestor. That is the most parsimonious or simplest way of explaining that. All right, now clades and evolutionary relationships result from phylogeny. So again, phylogeny is the study of the evolutionary past of a species. So how did they diverge or evolve from common ancestors? So simple explanation tells us that species that are the most similar are most likely to be closely related. And I'm telling you, the ability to sequence DNA has opened up a lot of crazy doors for people that study this. So the more DNA sequences that something has in common, the more likely it is that they share a recent common ancestor. So if I look at this phylogenetic tree here, okay, so I can see that a human and a chimpanzee share a much more recent common ancestor than do a human and an orangutan. So to find that common ancestor, I have to come all the way back here. Well, that explains why humans and chimpanzees have many more similarities in their DNA sequence than do humans and orangutans. Okay, these are much more closely related, have a much more recent common ancestor. All right, so how do geneticists and evolutionary scientists determine how many base sequences are shared between species? Well, what they could do is sequence all 3 billion human letters and then compare them to the billions of letters from, let's say, chimpanzees. But billion is a really big number, okay, and that could take a while. There's a much faster process called hybridization, and this is so incredibly cool. All right, so we're going to take one strand of DNA from species A, and we're going to separate it from its complementary strand. So I used to have 2 strands of species A. I'm going to take one of them. And then I'm going to take a strand from species B, another species, but this time I'm going to take the opposite strand. And I'm going to take these 2 strands and I'm going to put them together. Okay, so it should be this one. Okay, I'm going to put them together. So anywhere that there is a match, okay, anytime that a hydrogen bond forms between those 2 nucleotides, that must mean that there's a match, okay? Remember, because if there's an A over here, the only way that that can form a match is if there's also an A, and then on the strand that we're using, there's the complementary T. Okay, which would mean that they are both identical in that spot. So anywhere that there's a match would be a point in common. Anywhere that there's a difference, what's going to happen is those nucleotides don't like each other. So if this is an A and this is a G, they can't form a hydrogen bond between them. So they are going to repel and literally flip to the outside here. Okay, and so this is how we can tell how many differences there are in a single gene or sequence of DNA between two different organisms. Really cool stuff. So if we take a bunch of organisms and we use hybridization to determine the number of DNA base sequences that are different between those two organisms, we can then do a little bit of comparative work, right? And the more differences there are between two organisms, the less recent their common ancestor is. So if I have something with very few base differences, that tells us that we have a relatively recent common ancestor. If I have something with many more base sequence differences, that tells me that I'm going to have to go a lot further back in evolutionary time to find that common ancestor. And that's really a great way to put a cladogram together, because again, the whole point of a cladogram is to show evolutionary relationships. And so the more base sequence differences there are, the farther I have to go back in our evolutionary history to find common ancestors. Now, besides base sequences, we can use different traits to put together cladograms. One of the things we talked about previously is this idea of a homologous trait. So we talked about quite at length the pentadactyl limb, but there are others. Okay, and the only way to explain how multiple different organisms all developed that same trait or structure is if they have a common ancestor. So for example, eyes. All eyes, whether they're primitive, like these eyes on this scallop, or advanced, like this eye from a human, evolved from a common ancestor using pigmented cells. Okay, so all organisms have a certain amount of traits that they have in common with other organisms in their clade, again, came from a common ancestor. And we have to be very careful not to mix those things up with what we call analogous traits. So these are characteristics that might have the same function, but they don't have the same structure. Okay, so they did not come from a common ancestor. So the pentadactyl limb, for example, for a homologous trait, may have different functions like swimming versus grabbing, but they all have the same structure. Analogous traits like wings clearly have the same function, but very, very, very different structures. And the fact that those structures are so different tell us that they come from different common ancestors. So should not be used to put together a cladogram or to discern evolutionary relationships. So many of you, I'm sure, are already getting the hang of how to read these cladograms, but just to be clear, so they show how different clades evolved from a common ancestor. And so we've got a couple features here. We have these points of intersection called nodes. Nodes show a common ancestor. So turtles and humans have a common ancestor. It is whatever this organism was right here. So we had a single species that went through some kind of speciation event, okay? And eventually all of these other species developed, but the common ancestor is right here in the node. Okay, so we have a sister group, okay, or a clade, descendants of a common ancestor. So all of these came from this common ancestor. And then we have an outgroup, like the outlier, like the outcast, okay? And this guy comes from a different common ancestor, okay? Now, if we were going to include a different clade, like if I only wanted to talk about mammals, I could make my clade this group, and in that case, my node would be right here, and my common ancestor would be here, and then my outgrouping would be here. Okay, so it just depends on what you're considering to be a clade as to where to find your outgrouping and where to find your common ancestor. Okay, so if you think you're doing a great job with these clades, then you should be able to answer some questions on your own about this cladogram. So now would be a great time to pause and take a moment to kind of test yourself on how well you can read this cladogram. All right. And so if you're doing this at home with my notes, then here's an opportunity to check your answers. You can pause and see how you did there, but it's super important that you know how to read these and how to answer questions about them. So again, one of the great things about science is our ability and willingness to go back and use new technology to revise old hypotheses. And so because cladistics rely a lot on new technology, what we're uncovering is a bunch of new information about relationships. So in some cases, we thought we knew how things were related, but we have a ton of new evidence to suggest that there are different relationships that can be supported with DNA evidence. And so we're really in the process of changing how a lot of organisms are classified. Now, in a previous video, we talked about how birds are in a class all of their own, how we had the phyla of Chordata, and in that phyla we had things like fish, amphibians, birds, reptiles, mammals, okay, and we had some kind of classification that kind of seemed like that. Well, new DNA evidence and new evidence from the fossil record have told a very different story about birds. And we're finding that birds have a significant, like a super significant number of derived characteristics from a certain group of dinosaurs called theropods. There's supposed to be an O there. Okay, so for example, birds and theropods have a fused clavicle, that's their collarbone. They have super flexible wrists. They have hollow bones, which are good for flight. They have a very distinct hardened eggshell. And they have this really funky hip and leg structure where their leg points backwards. So whereas you and I, this is going to be embarrassing, if this were our front, here's an eye and a mouth, our legs would bend like this, okay? Birds, on the other hand, their legs bend the other way. So their hip and leg structure points backwards, and so do these ancient dinosaurs called theropods. So all of this evidence suggests that birds, modern day birds, are in fact an offshoot of very ancient dinosaurs, okay? And so where some of our other modern day reptiles evolved from other ancient ancestors, modern day birds probably evolved from those ancient dinosaurs called theropods. And I know this is kind of a hard idea to wrap our brains around, but this really kind of plays right into this law of parsimony, which suggests that if we have all of this evidence that tells us that they're so closely related, then we're probably better off accepting that simple explanation rather than the fact that these two just happen to have a lot of things in common, but that they didn't evolve from one another. And so birds, great example here of how we're redoing classification using new knowledge and new techniques. Okay, so that's super old school kingdom, phylum, class, order, family, genus, species, that taxonomy was pretty good. It's a pretty good system. Like if I know that something is in the phylum of mollusks, I know some things about it, like they're bilaterally symmetrical, okay, that they're not unsegmented, etc. But there are always some exceptions to those rules. So for example, most mollusks, like clams and oysters and such, have a shell, but octopus and squid, they don't. And so this way of doing those things kind of is a breeding ground for exceptions. Well, cladistics is a little bit different. Okay, here we're classifying things based on ancestry and evolution. And so if we're looking at a cladogram of mollusks, okay, perhaps it's something like this, where there is a common mollusk ancestor, but where shells develop and then we get branches for things like scallops and clams and oysters. Okay, so it gives us a lot more certainty in deciding who has what characteristics. And so while IB wants you to still know the general characteristics of each phyla or each class that you're responsible for, if you're on an IB exam and you get a cladogram, you should count your lucky stars, because once you have a cladogram, all you need to do is read it to figure out what some basic structures and other features that you're going to find in each group are. Okay, so I know that if I want to talk about bivalves, okay, I know that those are going to have all of these characteristics, all of these, all of these, all of these, okay, all of those are going to apply to this bivalve. The only characteristics that they're not going to have are on things that developed after they split off from the common ancestor. So a much more reliable way to discern traits about something. Okay, so let's say I'm some hotshot evolutionary biologist and I come up with a cladogram to show how various organisms are related in their common ancestry. So here was a node and a split, here's a node and a split, okay? So here's how I think that they're related in their ancestry. Let's say I do some deeper digging into DNA sequences and I find out that in fact, okay, A and B had a common ancestor, okay, that split much sooner than C, D, and E. Well, if I need to move something around to show a different pattern in the evolutionary relationship than what I was seeing before, that's okay. Cladistics is totally flexible that way, and that is something called circumscription. Okay, moving these evolutionary relationships around in a cladogram. So I cannot stress this enough. You can listen to this video and copy as many answers as you want, okay? But you're not going to get any better at this until you do some practice. So take the time to pause this video and do the practice problems on your own, and then you can use the next slide to check your answers. And I hope you're getting the hang of cladistics, and that'll do it for chapter 5.4.